Okay, so we left off by talking about isoosmotic, hyperosmotic, hypoosmotic, all right? So then the next slide is osmotic pressure. So osmotic pressure is a pressure derived from particles in the solution. These particles influence the movement of water. The side with more particles wins. You can click on the link and do the osmotic pressure activity. The trick to figuring this out is the greater the concentration of a solution, the greater the osmotic pressure will be, the greater the pull will be on water. So if you have a greater particle concentration, then that's going to give you a greater osmotic pressure, and a greater osmotic pressure is going to make water move towards it, right? Water wants to dilute the side with more particles, and because there's more particles in a higher osmotic pressure, then water is going to go to that side. Why is this important for our bodies? We have three compartments, right? Inside our cells, in the interstitium, and then in the plasma. And so the particle concentration of those different compartments are going to affect which way water moves. And so you want to be careful. So let me give you an example. Okay, so let me go over an example with you. So let's say we have a 0.9% solution and a 2% solution. Which one has the greater osmotic pressure? So to answer this, 2% is a higher number, so that's going to have a higher osmotic pressure than the 0.9%. To prove this to yourself, you can actually do 2% is 2 grams per 100 mil. 0.9% is 0.9 grams per 100 mils, right? And so which one has the greater unit of measurement? It's going to be this one. So the 2% has a higher osmotic pressure. The 0.9 has a lower osmotic pressure, right? So if we had a cell, 2%, actually, let's do it this way. Let me erase that. If we had a cell, so our body cells are 0.9%, always. The physiological range for osmolarity is 0.89% to 0.91%, okay? So let's say this is a red blood cell or a body cell, right? 2% on the outside, 0.9% on the inside. For our cells, the only thing that can move is water. Is that true in reality? No, but that's going to be true for right now. So which side has more particles? The 2% solution has more particles. So which way is water going to move? Water is going to move out of the cell into the 2% solution to dilute that 2% solution. So what's going to happen to the cell? The cell is going to shrink or crenate. Okay. Tonicity. So how do we use tonicity? Tonicity is a relative term that describes how cells are going to behave in a solution. This definition is not entirely true, but it indicates a concentration of the solution outside the cell. So this is how you're going to use tonicity. Tonicity really is the concentration of the permeant solutes, or the ones that can't cross the plasma membrane, but we'll use it this way because it's easier to understand. The cell is always 0.9%. And so we use isotonic, hypertonic, and hypotonic terms. Okay, so here's a picture of all of the different red blood cells. A red blood cell in an isotonic solution, so this is an isotonic solution. The red blood cell is 0.9, the solution is 0.9, right? The solution is the same as a red blood cell, so it is an isotonic solution. There's going to be no net movement of water occurring when red blood cells are in this type of solution. And so we like using isotonic solutions for IV fluids because you're not going to have massive amounts of water moving into the red blood cells or into your cells and blowing them up or shrinking them. Hypotonic solutions, right? Hypotonic solutions are solutions that have a lower concentration of dissolved solutes than the cell, right? So the cell is 0.9. So if you look at the red blood cells, the cell is 0.9 right here, right? Your cell is 0.9. And then the solution outside could be 0.5 or it could be 0%, right? So yeah. So if this is 0.5 and this is 0.9, which way is water going to go? Water is going to move into the cell and swell it, right? So the cell is going to swell in a 0.5% solution, and it's going to hemolyze in a 0% solution. Hemolyze means to blow up. And so notice water moved into this red blood cell and blew it up or burst it. A hypertonic solution means a solution has a higher amount of particles in it than the cell. So the solution has a greater amount of particles than 0.9%. Water is going to move out of the cell, and so the cell is going to shrivel or crenate, right? So a hypertonic solution, let's do 2%, would be hypertonic, right? Red blood cells 0.9, so water is going to leave the red blood cell and go into the solution. The greater the concentration difference is, the more water is going to move out, or the more water it's going to move out. Let's say this is 10%, right? So the cells are going to shrivel or crenate. Eventually they can crenate so much that they're not functional or that they tear because they've shrunk so much. Okay, so I want to take a second now to go over some situations. So let me see, get out a piece of paper, and I want to go over some stuff with you. Okay, so here's our whiteboard. So for the molecular activity lab, part C is tonicity. You're going to be using, or if we were doing this in lab, you would be looking at red blood cells in different solutions, okay? So let's write the solutions up here. So the 0% solution is right there. The 0.5% solution, 0.9% solution, and 2% solution. Okay, so the first thing is, do we care about the particles that make up these solutions? No. This solution, the 0.5, could be all sodium chloride. It could be 0.25 of glucose and 0.25 of sodium chloride. We don't care because we're just looking at the total particle concentration. Okay, so let's go over how to use osmolarity and tonicity. So let's work through these. Let's draw our pictures first. So here's your red blood cell, right? What's the concentration of your red blood cell? 0.9%, right? And then the solution that you're going to put it into is a 0% solution, right? This situation would be 0.9 inside the cell. Sorry, you guys, it's hard to write with this. And then 0.5% outside the cell. Let's do the eraser. Draw. Try this again. So 0.9 inside the cell and 0.5 outside the cell, right? For this one, it would be 0.9 inside and 0.9 outside the cell. 2%, oops, would be sorry, I did it again. .9 inside the cell, and then 2% outside the cell. Okay, so first I want you to describe these relationships using osmolarity, and then I want you to describe these relationships using tonicity. Okay, so you can pause it and work on those and then come back and check your answer. Okay, so let's go over the 0% description. So I can first describe this relationship by saying the solution is what? Hyper compared to the cell is hypotonic, or excuse me, well, hypoosmotic. Let's do osmotic first. Sorry. Compared to the cell, right? Then the next way to describe this is we can talk about the cell compared to the solution. So the cell, what is the particle concentration for the cell? The particle concentration for the cell is hyperosmotic compared to the solution. So you can write that as well. The cell is nope, sorry you guys. Is hyperosmotic. Then the next way you can describe this is using tonicity, right? So for tonicity, we're naming the outside solution. So the solution is what? It's less, so it's going to be hypotonic. Okay, do we need to say compared to the cell or relative to the cell? No, we don't, because when we use tonicity, we are comparing it to a cell. So the root of tonic means that there's a cell in there, right? So that's how you use tonicity appropriately. You're always naming the outside solution. You're assuming that a cell is there, and whatever solution you put that cell in, it's going to dictate what way water moves. So the cell is always 0.9%, and you're always naming the outside solution. Okay. Why do we use tonicity? It is a really great, fast way to explain what's going to happen. So if I give somebody a hypotonic solution, I know that it's less than 0.9, right? So I know that it's lower than 0.9%. And so that means water is going to move where? Water is going to go into cell. And what's going to happen to the cell? Cell will swell, right? Or blow up. In the case of 0%, it's going to blow up or hemolyze. In the case of 0.5%, that's not as great of a difference, so the cell's going to swell. So you can do, when you use tonicity, you already know which way water is going to go because you know the concentration of the cell. The concentration of the cell is not going to change, and you know what's going to happen to that cell, right? So 0.5% is the same. So the solution is hypoosmotic compared to the cell. The cell is hyperosmotic, whoops, compared to the solution. Did you guys catch that? Whoops. Compared to the You're probably thinking, yeah, I was confused on that. And then this solution is hypotonic as well. Okay, so these fit for both of those. Uh, which way is water going to move? Water is going to move in, and this guy's going to swell. Okay, in this situation, water is going to move in, and this red blood cell is going to blow up or hemolyze. Hemolyze means to blow up. The red blood cell blows up. In this case, that's why you don't inject pure water into somebody, because the water is going to move into the red blood cells and blow it up. You do use a 0.5% solution for dehydration. So if this cell is dehydrated, it looks like a raisin, you want net movement of water to go into the cell so it swells. All right, explain this setup. So one, the solution is what? So the solution is isoosmotic to the cell. The cell is isoosmotic to the solution, right? And then how do you describe this using tonicity? Good. The solution is isotonic. Sorry, guys. The solution is isotonic. Right. So if I put a red blood cell in an isotonic solution or isoosmotic solution, you know that the concentration of the solution is 0.9% particles, because the red blood cell is 0.9% particles, and so there's going to be no massive movement of water, right? Some water is going to move in, some water is going to move out, but there's going to be no change. What about 2%? So the solution, let's see, the solution is more or less or equal. Good, more, so that would be hyper, right? Osmotic to the cell. The cell is hypoosmotic to the solution, right? And then 3, you're naming this outside solution and it's more particles. So what do we call that? Good. The solution, is hyper. Perfect. Okay. Good. So this will help you for part C for the tonicity of the red blood cells. You can actually use the pictures in the lab, the pictures in the PowerPoint to help you with the lab, because obviously we can't look at red blood cells in lab because we don't have a lab. So you can use those, and this should help you as well. Out of all of these, which one has the highest osmotic pressure? Good, the one with most particles, this has the highest osmotic pressure. Which one has the lowest osmotic pressure? Good. Awesome. Okay. So let's go back to the PowerPoint. Okay, so for this one, let's see, you can use these guys as for your part C. So in your lab, part C is tonicity. This is your isotonic solution. So this is your 0.9% solution. This is your 2% solution. This would be a 10% solution, what you would expect to see in a 10% solution, even though you don't have to record that for lab. And then this would be a 0.5% solution, and this would be a 0% solution. Okay, so this is what it would look like in a 0% solution. So there's red blood cell fragments, and a 0.5% water moves in. The red blood cells look swollen. Isotonic, there's no net movement of water. 2% water is moving out, so the cell crenates or shrivels, and then 10% more water moves out, and so that cell really crenates or shrivels. It looks like a raisin. Okay, so go ahead and fill out your molecular activity part C based off of that. Okay, so hopefully that's clarified tonicity a little bit. Let's go over membrane transport, right? So diffusion is movement of a molecule from where it's highly concentrated, right, to where it's least concentrated. Osmosis is movement of water down its concentration gradient as well, or movement to the side where there's more particles, right? So water is moving to the side with more particles because it wants to dilute the side of more particles. Okay, so this is considered to be passive or simple, excuse me, simple diffusion when the molecule just goes down its concentration gradient. What molecules can do this? Go back to your slide, a few slides back. So molecules that can just diffuse through are lipophilic, right? So they're lipid soluble, or small and polar, right? So for example, benzene, water, ethanol, okay? They use simple diffusion. Osmosis is movement of water. Water's a polar molecule, it's just gonna go right through the plasma membrane. How do we get glucose and sodium and chloride in? So those guys can't go through the plasma membrane because this whole region is lipophilic. And when that whole region is lipophilic, there's no way you're going to get a charged ion through that fat soluble region. It's not going to happen. So we have to have a way to do this, and it's called facilitated diffusion. So you think of a little carrier protein or a little transport protein helping. So this side has more glucose, this side has less glucose. Glucose is going to move down its concentration gradient using usually a carrier protein. So if we're talking about facilitated diffusion, there's different types. So for a channel protein, one that's always open, like a little macaroni tube like this, it's usually going to be sodium or ions. For a carrier protein, that's going to carry larger molecules, it's usually going to be glucose. Okay. There are always exceptions to the rules, but just so you have an idea. Active transport is actually moving a molecule from where it's least concentrated to where it's highest. And in order to get molecules being shoved to where there's more of them, you're going to have to use ATP, right? Your body doesn't want to do this. This is taking molecules from disorder to order, or chaos to order. Your body doesn't want to do that. So you have to use ATP. And when you do that, it's called active transport. Okay, so let's look at facilitated diffusion. So facilitated diffusion is still passive. Why is it passive? We don't need ATP, right? So no ATP needed. So you don't need to expend energy to get this to work. It moves ions and large polar molecules like glucose down their concentration gradient. They can't they can't simply, or simple diffusion, right? They can't simply diffuse through the membrane due to the size or charge, so they need to be transported. There are two categories of transport proteins. The first one is channel proteins. These guys, some of them are non-gated channel proteins and some of them are gated. Out of the gated, there's ligand gated and voltage gated. So ligand means something pops open or something binds to that channel protein and pops it open. Voltage gated means something, you hit a certain voltage inside the cell and it pops open. And then there's carrier proteins. So let's look at those guys. So this is just a transport channel, right? Or an ion channel. So this is going to allow calcium through. Calcium is going from where it's highest to where it's lowest. So it's still going down its concentration gradient. Amino acid, this doesn't really happen, but that's okay. And then glucose is going down its concentration gradient, but it needs something bound to get this channel to open. So if something binds and pops this channel open, we call this a ligand gated channel, because that ligand has to bind to pop this open, and then glucose is going to go in. Typically, you guys, this is glucose has some type of carrier protein to get it inside the cell. But an example of this, to get glucose inside the cell, this would be insulin, right? So non-gated channels are always open. They look like little macaroni tubes. They're going to let ions go through the plasma membrane in any direction, but they're always going to go from highest to lowest. Gated channels, they're ligand gated channels. Look at the bottom left. This channel is closed, but once this ligand binds to the receptor on the channel, it pops the channel open and ions go rushing through, or whatever goes rushing through. Voltage gated channels, once that cell hits a certain voltage, that channel pops open, okay? And then for sodium voltage gated channels, they're actually open, then they become inactive, then they reset to closed. Then they're open, inactive, closed, open, inactive, closed. Carrier proteins are different. These guys are integral proteins. They're going to carry large non-polars, polars, or ionic molecules down their concentration gradient. Typically, this is going to be for bigger molecules, so amino acids, glucose, and proteins, right? How does it work? Those molecules have specific binding sites on that carrier protein. The protein is going to change its conformational shape to transport those ions or molecules, and it's going to resume its original shape after transport. Okay, so on the left-hand side, this would be glucose. So glucose is going to bind to the carrier protein, and then the carrier protein is going to kick it out on the other side. Notice we're not using ATP, so this is passive. You're still going from high glucose. You're still going from high glucose. Sorry, you guys. We'll do it over here. So we're still going from high glucose to low glucose, right? So you're still transporting down concentration gradient, so it's still passive diffusion. But glucose can't just diffuse straight through the membrane because it is large and polar, so you need a carrier protein, okay? Here is a carrier protein for, let's say, amino acids, so it works the same way. Carrier proteins exhibit the following characteristics, similar to enzymes. They're only going to bind a certain molecule, right? So the carrier protein is designed to bind, it's designed to bind one molecule, right? So if you have a carrier protein, if you look at the top one, that binds up an amino acid, it's not going to bind fructose, right? But if you have similar amino acids, they're going to compete for that binding site, right? And so carrier proteins display competition between or amongst molecules of similar shape. And these guys can get saturated like enzymes. So if you have, 100 glucoses, this carrier protein is going to plug and chug faster. But if you have 10,000 glucoses, this guy's going to plug and chug faster, but eventually it's plugging and chugging at its maximum speed or its maximum velocity. You can't get it to go any faster. And so we call this saturation. So it plateaus, so it's saturated. This just shows glucose carrier proteins. So you'll be looking at these when you look at the kidney, right? So here's glucose, it goes inside the cell. More glucose, it goes inside the cell faster. More glucose, faster, until these guys become saturated. Then they're still transporting glucose into the cell. They're just not doing it any faster. So we call that saturation. Active transport is when you take a molecule from where it's least concentrated and shove it where there's more of that molecule. Your body doesn't like to do that, and so you need to use energy to get this guy to work. So ATP is going to bind to this transport channel, split into ADP and phosphate, and when you break off that third phosphate group, you're going to release energy that's going to drive this pump. So active transport is pumping substances across the membrane against their concentration gradient. You're not going to get a molecule to magically go against its concentration gradient, so you're going to have to use ATP. Usually these guys are referred to as pumps. So you're pumping from low concentration to high concentration. An example of this would be sodium potassium pump. Okay, so primary active transport requires ATP. The example of this would be sodium potassium pump. So if you look at the picture, you can read the steps, but sodium is going to bind to this carrier protein. ATP is going to bind here. ATP gets split into ADP, adenosine diphosphate, and your inorganic phosphate group. When you break that bond, you release energy to this pump or to this carrier protein, and it kicks out the sodium, allows potassium to bind, and then drags in the potassium. So sodium potassium pump, how many sodiums does it kick out? Good, 3. And how many potassiums does it drag in? 2. And how many ATP do you use? 1, perfect. Secondary active transport is using the concentration gradient derived from a pump to make another concentration gradient to drive a pump. Right, your body's really good at this. It doesn't like to waste ATP. So your digestive tract does this, your liver does this. How do we get large molecules into the cell? So endocytosis is the very bottom. When a substance is brought into the cell, the plasma membrane invaginates or buds inward. Exocytosis is the membrane-bound vesicle carrying a substance fuses with the membrane and dumps the vesicle contents outside. Exocytosis would be a secretory vesicle. So let's say this is saliva or salivary amylase. So the vesicle with the amylase merges with the plasma membrane and dumps its contents out, so it releases the enzyme. This happens with mammary glands as well, and digestive enzymes by pancreas. Endocytosis is engulfing. There's several different types. There's phagocytosis, which is what a white blood cell does to a bacteria. So it engulfs it. There's penocytosis, and then receptor-mediated endocytosis. Penocytosis is moving particles from one side of a membrane to another. And then receptor-mediated endocytosis would be how we get cholesterol inside the cell, so that binds to receptors. Those receptors move in, invaginate, and make a vesicle. So this would be a vesicle containing cholesterol. Filtration. We would have done this in lab as well. So filtration works like a sieve. It depends on pressure differences on either side of a partition. Water is going to move from side of greater pressure to lower pressure. When do we use this? Blood pressure causes fluid movement out of capillary into the interstitium. The water and the small molecules move through, where the large molecules stay trapped in the blood. Other mechanisms would be secondary messenger systems, so cyclic AMP, G protein coupled mechanisms. The first messenger binds the surface of the cell, and that activates a whole secondary messenger system that activates protein kinase A. We talk about this, we don't talk about this so much in physiology, but you might talk about this in micro and immunology. Okay, that is it. You guys are done with this PowerPoint. Let's see.